before. Yeah, I forgot to press record. Okay, so now it's recording. So some factors that you absolutely have to know about your B cell development and maturation. Okay, so we're going to look at those, but I try to put some mnemonics that will help you to remember or associate, what are the factors and what they're doing. Okay. And then we're going to look in more details into your secondary lymphoid organs. So you absolutely have to know what is happening in the lymph nodes and what is happening in your spleen. Okay, you have to know about the zones, where the cells are hanging out, what they're doing, what's the functions of those organs. So that's mainly what we're going to focus on this lecture. Again, as you notice, I did upload the fresh PDF for today, which is just a combination of the other PDFs that I had up there, taking out some repetitive slides that I think it didn't make a lot of sense. Okay, so again here coming to the B cell development, okay? So we're going to look at those B cells and their presence at the germinal centers. Okay, what they are doing at those germinal centers and what are those germinal centers, right? Then you're going to look a little bit more on the interactions with those T cells and then on the activation of the B cells without the T cells, and that should bring you some connection with the material that was already in the last test about T dependent and T independent activation, right? And let's see here. All right, so why are we going through this process? Well, basically because it is very, very important to develop good antibodies to protect you from diseases, right? Remember that we had looked at MHC molecules and what kind of antigens that they can present. We also have seen that MHC genes are the ones that are most they have the most number of alleles in the population, and that's why it's so difficult to get a good match for MHCs. Like, for example, when you go for organ transplants, right, it's very, very difficult to get a matching of the 6 MHCs that you co-express. Remember, it's co-expression on at the genetic level. So here, I put this example here that during the COVID-19 pandemic, we know that a lot of people were developing antibodies against the virus, and some people were not quite able to develop antibodies, and those are the people that are more susceptible to the disease, and a lot of those people, because they did not develop antibodies on time and did not have a good immune reaction to the COVID, those are the people that actually died from COVID, which were a lot of people, right? I'm sure that everybody knows somebody who died from COVID, right? Okay, so we know that also not everyone that is exposed to the virus, not everyone is going to develop the disease, as well as, and also not everyone, on the other hand, not everyone is going to develop antibodies to the disease, right? So you have the people who are really immune and really resistant to developing the disease, and then you have other people who are more susceptible to developing the disease. And this is really, really interesting because the receptor for the virus that is in your lungs, right? The receptor for the virus is present in everybody. That's the ACE2 receptor, right? But it's still like some people will have different reactions and develop better antibodies than others. So here you're going to have a basic idea of how those reactions are taking place, and then how you can develop the antibodies that are effective and the antibodies that are appropriate for some type of immune response. Okay, so if you want to go and read the chapter, that's chapter 11. Okay, and what they're saying here, as we have seen before, you have this clonal selection hypothesis. Now, we have looked at how the B cell receptor develops, right? Remember, you have the pro-B, the pre-B, you have the heavy chain, the light chain, and all that stuff, right? And each B cell that is generated from your stem cell, right? Each B cell is going to have a very unique B cell receptor that is also your IgM, right, antibody on the surface. And once that IgM works, then you're going to have your IgM and IgD that are co-expressed, right? But each B cell is going to have a specific recognition for antigen. So you're going to have one B cell for a particular antigen, another B cell for another particular antigen. So when the antigen for a particular B cell shows up, that will signal the B cell to start proliferating, right? So this proliferation is what is called your clonal selection. So all the cells that are proliferating from this first stimulation here, at first, those B cells are going to have the same receptor on the surface, but then later, the B cell is going to differentiate further into other types of cells and into other types of antibodies that are expressed on the surface, right? So this B cell can differentiate after it expands, okay? It can differentiate into antibody secreting cell, and that's your plasma cell, or it can differentiate into a memory cell, and again, we have seen that before, right? But in the meantime, depending on the stimulus that this cell had, in the meantime here, something is going to happen that is going to change how that antibody works. It's going to change the affinity of the variable region of the antibodies, okay? And there is more to that story. So here we have seen that before, that each of the heavy chain locus, so this is, remember, the heavy chain is what tells what type of antibody that is going to be produced, right? So the heavy chain, the part that anchors it to the membrane, that is what's going to tell you if you're going to have IgG or IgM. So remember those heavy chain genes, those are called gamma, alpha, mu, epsilon, delta, right? And you have several deltas, and you have here like delta, here is showing, several gammas. Delta, you only have one for the D. For the gammas, for the IgG, you have here Ig or gamma 3, gamma 1, and then you jump something, and then you have gamma 2. You have several of the, of the gamma genes, because you have several types of G antibodies that you can produce, right? So here is saying that for every heavy chain locus, you're going to have 2 exons at the end of the locus. You're going to have 2 exons. They can be alternated during splicing. So that's differential splicing, right? So they can alternate between the secreted form of the antibody, the one that the plasma cell is shooting out, and the membrane-bound form, the thing that stays on the surface of the cell, okay? So here, those exons are those little pieces of genes at the end of each exon. So you have secretion exon, or you have membrane-bound exon. So here, due to alternative splicing, you're going to have antibody that is shoot out or antibody that stays on the membrane. That means the antibody here, after your B cell has seen antigen, this antibody here, if the cell was stimulated by a T cell, right? If it was T-dependent activation, then you're going to have here not IgM, but you're going to have some of the other types or isotypes of antibodies, okay? You have IgG, IgE, something else that can be on the surface of that cell, okay? Remember that in order for you to have that class switching, you need to upregulate or activate that enzyme that is going to do the class switching, and that is your AID, right? We have seen that before too. So here you're showing a T cell. So this is your T helper cell, right? Remember, this is what kind of CD molecule is there? CD4 or CD8? The CD4 for T helper cell, okay? So this T helper cell is going to interact with your B cell. And now, besides activating your AID, the T cell is going to tell your B cell what kind of antibody it has to shoot out, right? So the T cell is going to produce some cytokines. They're going to tell the B cell how to do that switch, right? So here, okay, we have seen that. I just mentioned this. All right. So that switch and further refining of your antibodies that really have to do with appropriate responses or appropriate production of the antibodies, right? So how that's going to happen and where that's going to happen, this is what we're going to look at now. So that's going to happen here in your secondary lymphoid organs, okay? And there are specific places where that's going to happen and specific type of stimuli that are going to allow that to happen. Okay, so just to review what is happening with your B cell, I'm sure that everybody's really familiar with this, right? You know that they are derived from stems, the pluripotent stem cell, right? So when you are in the bone marrow, which is showing here, when you are in the bone marrow, you have some structural cells in the bone marrow. So those are stroma cells, right? They are producing certain cytokines and they're interacting with your precursor cells, right? And after the differentiation, after the rearrangement of your B cell receptor, you're going to have your IgM. Then you're going to test that IgM. What do you call the type of testing here in the bone marrow for antigens, whether the cell recognizes self-antigen or not? Okay, you got 50% chance of getting it right. That was the wrong answer. It's called negative selection. Okay, so here in the bone marrow, if you recognize bound antigen, okay, bound what? On the surface of some cell, if you recognize a self-antigen, then this cell is going to die. But there is more to that story. Okay, this is just the beginning. Okay, what is showing you here in the bone marrow is that your B cell that recognized surface-bound self-antigen, look, this surface here has several self-antigens. So it's doing a multi-antigen type of crowding of those receptors, right? So this is when you have more than one stimulus that is activating that cell. When this happens against this bound self-antigen, then most of the time the cell will undergo apoptosis. But that cell, that B cell, can do another trick. It can rearrange the genes in the light chain to try to not bind to the self-antigen. Okay, so that's a process that only happens with the B cells. It's not going to happen with the T cells. Okay, so now let's see here that the cell passed. So that is not in this slide yet. So the cell did not recognize self. Now it is going to upregulate a little bit of IgD and is going to go to the periphery, right? Now, in the periphery, other processes are going to go on because you really have to check for self-reactive Bs, okay? So you're going to have to go through a process of peripheral tolerance that is going to happen in the secondary lymphoid organs, because you really want to make sure that you are not going to attack yourself. Okay, so we're going to look into that as well. Okay, so after that, of course, when you get activation of the B cells, you're going to produce the antibodies, and you're also going to produce memory cells. So you're going to produce plasma cells that are short-lived only for the time that you need your antibodies. That's the other thing to keep in mind, like how short-lived are they? When do they die? When is it that you don't need them anymore, right? You're going to keep that in the back of your mind because that's another important factor of B cell, okay? And after these cells are done producing the antibodies and they die, then, you know, they're gone, but you always keep a memory cell that can, again, be transformed into that plasma cell, okay? Okay, so now, important things that you have to know about the development of that B cell that I had not mentioned before, right? When you are here at the early pro-B cell stage, right? So remember, this happened also with your T cell development. So you have here multipotent or pluripotent stem cell that is deciding to go into your lymphoid lineage, right? So you have your common lymphoid progenitor cell, and the important thing for that is your interleukin-7, right? But this cell here, it can go into a T cell, or it can go into a B cell, or it can go natural killer cell, right? So it's lymphoid lineage. So here you have your interleukin-7 receptor. But now, since this cell is going to go and become a B cell, other things are happening, okay? You are going to have interaction with adhesion molecules in the bone marrow. Okay, so adhesion molecules are in the stromal cell, okay? Once the interaction, especially with this VCAM1 molecule happen, then this cell here is going to start upregulating certain markers. They are going to make sure that it's going to develop into a B cell and it cannot shift to a T cell anymore. Okay, so what are those that are important here? Okay, one expression that is important for lymphocyte lineage is this receptor tyrosine kinase. Is called FLT3. Okay, I'm not going to ask you about this, but it's good that you have heard it somewhere in your life. Okay, now here's some chemokines acting with it. Okay, here is your IL-7. I did ask you about that before. Okay, and here, let's see, it's a progenitor. Okay, this guy here, receptor tyrosine kinase KIT. Remember this guy, KIT, right? So this guy here is very important for the development of the B cell at the pro-B stage, okay? But then at the same time, you don't have only KIT that is being expressed here. You have these other factors like Icaros and these guys here. These guys here are important for B cell lineage commitment, okay? So at first you have your KIT, or right, the receptor tyrosine kinase that gets upregulated. The other name for this is CD117, but who cares, okay? And after this, you're going to have the interaction of the KIT with a stem cell factor that is on the stromal cell, okay? And that is going to cause proliferation of this B cell right here. Now look, the B cell does not have receptors yet, okay? It's just getting committed to a B cell. Besides that, you're going to have expression of other factors. Look at this. You see this guy here? EBF, EA2, PAX5. Those are very, very important for commitment of your cell to the B cell lineage. Okay, so you have early B factor and EA2. What those factors are doing, they go straight into the DNA and they push some of those histones around. They're interacting with the DNA. So they're doing histone rearrangement in order to expose the genes that have to be reoriented for expression of the B cell receptor. Okay? Now, after those factors are already pushing the histones, you remember what the histones look like in the nucleosome, I hope, right? Okay. So after that, then you get expression of this guy here, PAX5. And once PAX5 is expressed, then the expression of non-B cell genes, it gets suppressed. Okay, so the cell is definitely committed to becoming a B cell. Okay, now, things that are important to know here. Okay, what's going on here? I don't like that. Okay. I forgot you. So one thing that is important to know here is that once you have this KIT interaction with SCF, the thing starts to proliferate. Okay, the cell starts to proliferate. It's still without the receptor. So I put here this kitty cat and I say, you know, proliferation of kitty cat, you get a lot of kittens, right? So it's still in primary stage, you get a lot of kittens, but then later on, when you are at the late pro-B to early pre-B stage, this is when you're going to start getting the M spikes, Okay, and this is why I put here you transformed from a kitten to this beast with M spikes on the surface. Okay, so this is like production of kittens transforms into a beast with the M spikes. Okay, so here is of course total commitment to production of your B cell receptor. Okay, so here of course you have seen that before, right? And something else that that I have added here, again, when you get rid of the cells that are self-reactive, again, that is called central tolerance, right? Now, after that, the cell still has to undergo further testing once it gets out of the bone marrow, because in the bone marrow, remember that the repertoire of antigens, of self-antigens that is presented in the bone marrow is not as amplified as what you get for the development of the T cell in the thymus, right? Because remember, the thymus has that very special transcription factor, that is the ARI transcription factor, right? That really presents a lot of self-antigens. But then in the bone marrow, that's not going to happen. So some B cells that are self-reactive, they manage to escape the bone marrow, but they are mature. They have the IgM and they have the IgD. But then when they get to the lymphoid organs, and most specifically in the spleen, then they are going to undergo further maturation process. So they're going to be exposed to other self-antigens that they had not seen in the bone marrow. So that is called peripheral tolerance. Okay, so they're going to be further taught on how to react in the spleen. Okay, now good functioning of both of these systems is really, really important to avoid your B cells from producing self-antibodies. Right? If those selections are not very effective, then you can end up with diseases such as rheumatoid arthritis or lupus. And keep in mind that most of the diseases that are associated with self-antibodies, you have a function that the self-antibody blocks a certain organ function, right? Or interferes with proper communication of the organ. Most of the autoimmune diseases work like that, but not all of them. All right, so here, what's happening here? In the bone marrow, right? So when you have your multivalent type of self-antigen, right? Most of the time here, your B cell is going to be eliminated, but sometimes the cell can undergo receptor editing and then be tested again, okay? Now look at this, in the bone marrow, besides the multivalent type of antigen exposure, you also have soluble self-antigens. Okay, so when you have soluble self-antigen, look what happens. It's going to be more difficult for the cell to be activated because that soluble antigen has to cross-link at least 2 of those receptors. Here is easy to cross-link more, but here you are not really going to get good activation of your B cell with those soluble antigens. So what happens here is that the B cell is allowed to migrate, to come out of the bone marrow and go to the periphery, okay? But because these cells basically had one signal, most of the time those B cells are going to be useless. They're going to become the zombie cells. There's the zombie, right? So they are anergic, so they're not really useful. I don't know why they're really produced. They're part of energy. Okay. Okay. All right. So when you look at soluble antigens that didn't really activate any of the receptors, so those are the cells that are really important to also go to the periphery, and they have more or less the same type of function as the anergic cells, but they're not classified as the same thing. They are called ignorant cells or clonally ignorant cells. They can be activated, but they're not going to proliferate. So this production of those zombie cells, they really have to do with recognizing soluble antigens. Okay, soluble antigens can make zombie cells. If they are multipotent or multivalent antigens, that's when you can get either apoptosis or receptor editing. All right, so let's see what happens here. When you are undergoing that negative selection, right? So you can die, you can become a zombie, or you can change your receptor. So the editing of the receptor is only going to happen at the light chain, okay? And then after that, the cell will be able to migrate to the spleen and undergo further exposure to other self-antigens. Okay. Now, what is the percentage of the cells that will actually be able to leave the bone marrow? Look at this. 90% of the cells do not leave the bone marrow. So 90% of the B cells that you mate to the receptor, they die. So we're only talking about 10% of cells that are going to go out to the periphery. Okay, so that's a very, very small number of percentage of cells. Okay. Right, so again, what is the periphery? Anything that is not the thymus or the bone marrow. Everything else is periphery. All right, so this is a part that, yeah, it's kind of busy, but you do have some important information here. It's kind of concentrated, very busy, important slide. Okay, so let's go here step by step what is happening here. Once those B cells go to the spleen, let's do this thing. Once they go to the spleen, several things can happen to them. Okay, so this here, you have your immature B cell that was in the bone marrow. It went to the periphery and then it went to the spleen. So this is happening here in the spleen. So this part right there. Okay, so what is happening in the spleen? In the spleen, this cell is going to interact with some factors and is going to upregulate, is going to express a specific cell receptor on the surface of this B cell. So you can think of this receptor as the B cell best friend, because without it, the B cell is going to die. Okay, so if the cell has upregulation of this B cell friend forever, okay, B cell best friend forever, okay, the proper name for it is B cell activating factor belonging to TNF family receptor. Who cares? Just think about your best friend forever. That should tell you this thing is going to keep your B cell alive. Okay, so when this thing goes to the spleen, goes to the follicles in the spleen, because this is where B cells like to hang around, as we're going to see. Okay, so it's going to upregulate this best friend, and this thing has to be stimulated in order for the B cell to be alive. If you do not stimulate this receptor, the cell is going to die by apoptosis. Okay, so this is happening in the spleen. Now you can imagine that if you have some people that do not express their best friends, you cannot keep those B cells alive. Okay, so those B2 cells that went to the spleen and did not get a survival signal, those B cells are going to die. So that means the person is not going to produce antibodies that are dependent on class switching on the secondary lymphoid tissues. Okay, the person is going to be immunodeficient, and this is a type of primary immunodeficient disease. Okay, because you're born with it. You're born without the gene, so you cannot really produce the antibodies that you need to defend yourself from a lot of stuff like viruses, right? Or even some helminths when you need IgE class switching. If you need IgG class switching, you're doomed, because those cells, they're going to die. You're not going to have them. Okay. On the other hand, if you have too much production of BAF, Okay, too much of a best friend, that's not good either. Then you can have some autoimmune diseases. And a classical example of this type of autoimmune disease is, okay, I gotta pronounce that thing. Sjogren's syndrome. Anybody volunteer to pronounce that? Okay, everybody at once. 1, 2, 3. Sjogren's syndrome. What is that? Well, it is a kind of complicated syndrome with too much production of antibodies. Okay, so this is an autoimmune disease. Okay, so if you don't have it, it's a problem. If you have too much of it, it's a problem. Okay, so you have to have the certain amount or the certain level of your best friends around. Okay, okay. So this is what I mentioned. Now, that best friend and survival of this type of B cell, which is the B2 type of cell, this thing is happening at your secondary lymphoid tissues, like here is your spleen, right? And that is going to be important for survival of the cell. That's going to go to the lymph node, and then the cell can get activated, right? So the cell can become a plasma cell in the lymph node, but the cell could also be activated in the spleen and become a plasma cell in the spleen, and then do the class switching and all the other stuff, right? Besides that, you do have another line of these cells that can become follicular B cells. Okay, so this is another type of differentiation of your B cells. Now, this has to do everything with your B2 cell type. Okay, so when you're talking this thing, you're talking B2. Okay. Besides that, remember that you also have another type of B cell that is the B1 cell, right? And that is mainly related to innate immune system, as we're going to see in a few slides down the line. So this is very important for B2 further development. Okay. All right. So again, when you have your naive but mature B cell, remember that's your B2 cell type, right? Remember the two receptors at the surface and everything that I talk here. So this is for T-dependent activation. And then here you have those receptors that are important for T-independent activation. We have seen that before, so just to refresh your mind. Okay. So here, besides your B2 cell, right? That this is what happens to your B2 cell, right? Okay, about the timing of your B2 cells. If the antigen for your B2 cell, developed mature B2 cell, if the antigen is not found within a period of time, which is actually less than 2 months, then your B cell is going to die. So that B cell that is specific for that antigen is going to be gone. Okay? Does that mean you're going to be deficient on a cell that is going to recognize that antigen? No, because your bone marrow is continuously producing new B cells. Okay? That one is going to be gone, and then no one's going to be produced. Now, besides those B2 responses, remember you have the other type of B cell that is your B1 cell type. Remember I mentioned that the B1 cell is produced during the fetal life, so before you're born, and the organ that produces this B1 cell is the liver, okay? And then those B1 cells, they are really part of your innate immune system, although they are lymphoid cells. Okay, so what happens here is that those B cells, they're going to be hanging around in these areas that is called peritoneal and pleural cavities. So now is a refresher on your anatomy courses, right? So peritoneal cavity is here showing that is around your abdominal muscles, and the pleural cavities are really surrounding your lungs, right? Do you remember learning about those membranes and the organs that they are, encompassing? No. Okay, so here I'm telling you what those are. Okay, now those B1 cells, the way that they get activated is by T-independent response. Okay, and they are going to produce what kind of antibodies? IgM. Okay, they are going to produce IgM antibodies. So when you see a patient that has plenty of IgM antibodies and does not have any of the other isotypes, so you could start thinking, is it a problem of AID production? Is it a problem of the best friend production? I mean, it could have several factors, but those are two factors that are going to really hinder the class switch of antibodies. Okay, so now back here to the B2 cells, right? Because those are the ones that are T-dependent, right? Remember, those can also be activated independently of T, right? So those are going to go to your lymph nodes. They're going to go to the B cell zones, okay? And they are, of course, different than your B1 cells, right? How can you tell them apart? Well, they have a lot of the same receptors on the surface. They have a lot of those B cell receptors besides the CD19, right? CD19 is present in both B1 and B2 types, right? Now, a major difference between your B2 and your B1 cell type is the presence of this CD21 on the cell surface. Now, CD21 is a CR receptor. I believe it is CR1, okay? I believe it's CR1. Okay, this is one major difference. All right, so let's see what happens here. When you have your B cell and the B cell goes to your lymph node, so it likes to hang around in a B cell zone, and that place where it likes to hang around is in the follicle, okay? So you call the B cell a follicular B cell right here, follicular B cell, okay? Now, the B cells in their B cell zones, they are going to wait for their antigen to show up. So they sit there and just wait for, something to bind to the receptor. So it could be different types of antigens. It could be tiny antigens, it could be big antigens. Remember that your B cells are going to recognize 3D, so they go recognize the whole folded proteins, right? The whole folded thing. So how is the antigen going to get there? Well, the antigen can come from the lymph, right? From lymphatic circulation. And see what happens here? You're coming here from the lymphatic. These are the afferent lymphatics for arriving. And if the antigen is small enough, it gets filtered through these little channels. They're called conduit channels. And they end up in this follicle, okay? And if they're small enough, they can go and interact with the B cell and activate the B cell. Now, this type of activation is T-independent, okay? And this is happening in the follicle. Now, on the other hand, if you have a big chunk of antigen that it doesn't go through these tiny little spaces, you can have either a macrophage that can carry it to the follicle area, or you can have some dendritic cells that can bring it into the follicle. Now, these dendritic cells are going to bring the antigen, big antigens, to the follicle and present it for the B cell, right, to start to differentiate. Again, these guys are not T cells, they are dendritic cells, okay? So here you're going to have T-independent differentiation. So this B cell here is only going to be able to produce IgM antibody, okay? So when those antigens get in there and the cell gets activated, you're going to have the clonal selection, and these cells are going to present IgM, is going to shoot out IgM antibody. Remember, IgM is a pentamer, right? Now, after that, you can have, right, meeting of your activated B cell with your activated T cell. When your T cell gets in there and interacts with the B cell and is able to do the class switching, right, then your B cell is going to start to do the affinity maturation of the receptor. Now, this is when the follicular dendritic cell is going to be involved. So in this picture, you see here two types of dendritic cell. You see dendritic cell outside, dendritic cell in the follicle. Now, these guys here are not going to, at first, present antibodies, present antigen to your cell. But what they're going to do, they are going to interact with antigen experienced B cell, and they are going to start fine-tuning the binding of that receptor. They are going to do the affinity maturation of that receptor. So the follicular dendritic cell is what is responsible for activating receptor affinity maturation. Okay, I know there's a lot of words on what I just said right now, but if you are half asleep, make sure you go back and you understand what this means. Okay? All right, so let's see what is happening here. This is what I just mentioned before. Small soluble antigens are going to go there. They go by the conduits, and they can activate the B cell. Larger antigens are captured either by macrophages or by dendritic cells, and they can be brought to activate your B cell in the follicle. All right. So here for your B cell activation, see CD21, that is actually your CR2. Remember the guy CR2 recognizing C3D? Okay, so this guy is a CR2. I always make confusion in my head between CR2 and CR1. So these guys here are typical of your B2 cell subtype. Okay. All right. And let me see here. What else is important? Okay, we already mentioned that. Okay. Okay, also second signal, remember, can come from a TLR, recognizing PAMs. All right, so what is happening in that follicle, right? So B cell is sitting in the follicle, that's the B cell zone, and is going to interact with the antigens. They are going to pass through all these stromal cells. Again, stromal cell is a type of structural thing of the lymph or any organ, okay? So stromal cells are all around, and the antigen keeps on, bouncing around until it gets to the zone for B cell activation, okay? And once you have your B cell that recognizes that antigen, it's going to proliferate, okay? It's going to proliferate, divide, and it's going to start forming a germinal center. Okay, so now is the formation of that germinal center. At first, you're going to have a primary germinal center, primary follicle, and then you're going to have a secondary follicle because the thing is going to grow. It's going to get bigger. Okay? So let's see what is important here. All right. So this is starting to look at your lymph node. Again, you have here your cortical region. This is where your B cell is located. You're going to have a region close to the cortical region. There's your paracortical region. That's where your T cells like to hang around. And then you're going to have a medullary region that is around here. And mainly going to find macrophages over there. Okay, so let's see here what happens. All right, so this, I believe you have seen this slide before, right? When you have T cell activation of your B cell, depending on what kind of cytokines your T cell is going to produce, is going to show to the B cell, you're going to have the class switch, right? So in this case, if your T cell produces interleukin-4 and interleukin-5, you're going to do the class switch to produce IgE. You're going to see later why, okay? If your T cell produces interferon gamma, you're going to do class switch for IgG. Well, this G is for gamma, or gamma is for G. Okay, that's easy to remember. Okay, so what is happening here in that follicle? Okay, so looking here in the follicle, your B cell gets in there. This is your conventional naive B cell, B2 type, right? So what can it do? It can go inside of the follicle, okay, and sit there forever, wait for the antigen. Now let's see that your antigen has showed up. Okay, your cell is activated, and now you have T-dependent activation. Okay, and what is going to happen here? First, you're going to have proliferation of those cells, and inside of this germinal center, you're going to have interaction with a follicular dendritic cell, okay? That is going to improve the affinity of your antibody, okay? So inside the germinal center, you're going to test whether your antibody is good or not. If your antibody has low affinity, the cell is useless, so you're going to kill it, okay? You're going to kill the cell that has low affinity. If you have an antibody that for some reason is going to be reactive, is going to react to something of self, then the thing is dangerous, so you're going to kill that cell. So only the type of cell that is producing an antibody that is useful and not very low affinity and not self-reactive, that antibody is going to start to be changed by stimulation of the follicular dendritic cell, and you're going to have better antibodies that are going to get produced, okay? All right. Okay. Okay, so I mentioned that. Okay, I just mentioned that too. Dendritic cells, antibody affinity, okay. All right, so important thing about those follicular dendritic cells, okay? They are going to produce a couple of cytokines that are important for B cell, for their maturation, right? And those follicular dendritic cells also produce that BAF, okay? They also are going to stimulate that receptor on the B cell to keep it alive, okay? So let me see what else is important here. So what is happening in the germinal center? You're going to have affinity maturation. You're going to have class switching, okay? And you're going to generate those plasma cells that are better than the original plasma cells that you generated. So the plasma cell with high affinity are going to be formed there. Besides all that, you're also going to have formation of memory B cells. And those are going to go to a couple of places. Some of them are going to hang around in the periphery, and some of them are going to go back to the bone marrow and sit there until they are called upon the second activation of encountering with the same antigen. Okay. All right, so let me see here. All right, so the zones of the follicle. What is happening here in these three different zones of your follicle, and once the B cell is activated, you call that a germinal center. Okay, so follicle and germinal center is the same thing. Basically, the follicle is without activation, and then it becomes the germinal center if the B cell gets activated. Okay, so you have different zones here. So look at this picture here. You have a zone that is looking darker. You have another zone that is looking lighter. So different things are happening in these different zones. Okay, in the dark part of this germinal center, okay, you are going to have proliferation of your cell, and you're going to start to have mutation of your receptor. That's what you call hypermutation. Okay, so proliferation and hypermutation of the receptor. Here, after proliferation and hypermutation, you're going to start testing the antibodies to see if they're useful or self-reactive. Okay, if you have a good antibody, if you have a good cell that produces good antibody, is going to go to the light zone and is going to be further developed by interaction with your follicular dendritic cell. Okay, so the light zone, you're going to have hypermutation, you're going to have affinity maturation, and what is the proper name for it? No, I forgot the proper name for it. But basically, you're going to have affinity maturation of your receptor, okay, in the light zone. So dark zone, thing is multiplying and changing receptor. Light zone, receptor gets better. Besides all that, you're going to have another region that is right around it, and it's called the mantle zone. Okay, in the mantle zone, that you see plasma cells here, you're going to have secreting cells, right, secreting plasma cells, and you're going to also have formation of your memory cell. So this area around the germinal center is called your mantle zone. Okay, so 3 areas: dark zone, light zone, and mantle zone. And you should know from these things, what are good test questions out of this? Like, in which zone do you have affinity improvement or affinity hypermutation of your B cell receptor? And you say, oh, that thing happens in the light zone. Where do you have formation of B cells? Oh, that's around the outside. That's a mantle zone, something like that. Okay, so here, this is from Lecturio, and is also showing, proliferation of a B cell in the dark zone, and then affinity maturation here in the light zone, right? And then in the mantle area, your memory cells and plasma cells. Okay, so these I had mentioned before. You can read that. And here is just to show you, where your memory cells go. They stay circulating. Okay. And this is just a review of what happened with the secondary response to the antigen that was seen before, right? Remember, because you have those memory cells that are ready to start shooting antibodies, you're going to have a much higher response and much faster than your primary response. Okay, and here's a little summary of what is happening with your B cell activation. Okay. Now here, going to the secondary lymphoid tissues. Okay, so more details on what is happening and the structure and function of those secondary lymphoid tissues. What are the cells you're going to find there? Well, same cells we have seen before. Okay. And this is from Lecturio. Again, we have seen this before. Okay, and this is from your book. You can read that. Okay, so here, getting into the structure of your lymph node. So you absolutely have to know what the lymph node looks like, what your spleen looks like, and you have to compare the zones and compare the functions. Okay. So the cells that are making the capsule and the structures that hold this thing together, those are called stromal cells. So stromal cells are for support, and you can think of stromal cell as structural cells. Okay, so this stro, it kind of sounds the same. Okay. So here you have the capsule. It's a hard capsule on the outside. Okay, and then you're going to have also some indentations of the capsule that are not quite showing here, but you're having the second slide here. Okay. What you're going to see here, you have B cell zones and T cell zones, right? So I don't know why this thing shifted right away, but it's the same type of idea anyway. So B cell zones go close to under the capsule, and then T cell zones are very close to the B cell zones because they have to have an area in which they are going to interact. Okay, so T cells are gonna hang around the T cell zones. When you look at the T cell zone, you're gonna find a lot of reticular fibroblastic reticular cells that are arranged there, and the T cells like to interact with these reticular cells. Okay, they like to sit around there and interact with dendritic cells and just go through and not just flow through very quickly. They are held at those fibers. Okay. So those are the fibers. Let me see what's important here. Oh, finally, your first question. Okay. Okay, the question, which of the following is the main site of activation of the adaptive immune system? Oh, okay, that's so easy. Okay, so you shouldn't take more than like 2 seconds to answer this question. Those are just like the 3 points to make sure everybody's awake. Okay, so 5 seconds. So somebody is semi-awake. Should I give you more time to change your mind? Okay, what's your answer? Hey, you're going to activate them in your secondary lymphoid tissues. Here, no, not D, just A. Okay. Now this thing got stuck. All right, so structure of the lymph node. You have your cortex. So here you're going to find your B cell zone. You have your paracortex. You're going to find your T cell zone. And keep in mind, if the thing starts with P, that's where your T cells like to hang around. So paracortical area, same thing here. You're going to see in comparison to an area of the spleen. Okay, and then in the center here, you're going to have the medulla, and you're going to find a lot of spaces here. They're called the sinusoids. Okay, and here you're going to have macrophages that like to hang around. So here are your afferent vessels, and here is going to be your efferent vessels. Okay. So what is important to see here? Remember that cells can get into your lymph node in one of two ways, right? You can go from the lymph, or you can go from those high endothelial venues, so either from lymphatics or from the blood. Okay. Okay, so looking at the lymph node, you're going to have this hard capsule, and this is called collagenous capsule, so it's a tough capsule. And the capsule, the tissue that makes the capsule extends to the inside of the lymph node, and this is what you call the trabecula. So the trabecula is going to subdivide this lymph node into things that look like pizza slices. Okay, so pizza slices here, the little trabecula. Now, under the capsule, like right there, you're going to have a space. That little space under the capsule is what you call the subcapsular sinus. Sinus for space, okay? So let me see here. The inner part of the lymph node is what you call the medulla, okay? And the medulla has those spaces that you call medullary sinus. Okay. Again, your B cells like to hang around those follicles, and those are in the area of the cortex. When the follicle, when the B cell in the follicle recognizes antigen, then the follicle becomes a germinal center. Okay, germinal center only if it recognizes antigen. Okay. So what is going to happen in these zones? You're going to have activation of the cell, and if the cell stays there, it's useless. So after activating, the cell is going to come out and go to the site where it's needed. All right, so looking here at this spleen, it's right there close to your stomach. Okay, so the spleen is right here, and it is your largest lymphoid organ, right? So what is happening here with the spleen? You're going to have this big artery, the red one, okay, splenic artery that goes in there, and coming out of there, you're going to have this splenic vein. Now, the way that this thing is working inside the spleen is a lot different than it's working everywhere else in the body, okay? Now, the place that you have both of the artery and the vein that are entering or leaving the spleen, this part here you call the hilum, okay? So this, like bottom part of the kidney bean shape, this is the hilum. Okay. Now, what is the spleen good for? It's going to filtrate the blood, so it's going to look for parasites in the blood, but that's not the only job of the spleen. It is also going to store platelets. Okay, so it's a source of platelets, and is also going to get rid of damaged blood cells, red blood cells, or tagged red blood cells, opsonized red blood cells. It's going to get rid of those, and you're going to see here how it's going to happen. Okay, so areas of the spleen. You have a very strong capsule, okay? And also from the capsule, you have the trabecular, just like you had with your lymph node, okay? So you have the subdivisions. And then you have a different type of organization here, okay? So before I go into organization, I'm just making sure I cover this here. Okay, damaged red blood cells, they are disposed in the spleen. If you have bacteria or viruses in the blood, they get trapped in the spleen. If for some reason you don't have your spleen, then you're more susceptible to infections, like bacterial or virus infections, right? Although the rest of your immune system and your lymph nodes should take care of that, but you are more susceptible to infections. All right. So besides of that, you can reserve some blood in the spleen just in case you need it when you're exercising very vigorously or you need some blood for any other reason. You're going to store also platelets and some monocytes in case you have some infection and you have to liberate monocytes. Okay. So areas of the spleen here. So defense against blood pathogen. So you have mainly two areas in the spleen. You have the red pulp and you have the white pulp. Okay, we're going to look into those areas here. Okay, so let's see. The artery that goes into the spleen, that is your main artery right there, called the trabecular artery. And then it's going to be subdivided here into a central arteriole. So artery is a big one. When it branches out and becomes smaller, it's called arteriole, right? And then the arteriole is going to branch again into follicular arterioles. So central arteriole goes in the middle. Follicular arterioles are branching out from that one. Okay. In the center here, this is where you're going to have your white pulp. Yeah, here is darker, but that's where you're going to have your white pulp. And on this side, you're going to have your red pulp. You have better pictures of it. So let me go to the following one. Okay, I like this video, so I put the link here so you can go back and watch the video. Really, really, really informative. All right, so you have the capsule, you have trabecules, and then here's a splenic artery, okay, central arteriole, okay. Now, here, you're going to see this sheath, okay. I know I have a problem pronouncing that, but here you have a lymphatic sheath, and here is where your T cells like to hang around, and also some macrophages, okay. Now, when you look here, you're going to see that the lymphatic sheath is where you're going to have your white pulp, okay? So this is going to be close to, like stuck to your arterioles here. This is where you're going to have your white pulp. Around that is where you're going to have your red pulp, and there is a reason for that. Okay, so what is happening here? You have the periarterial, you have the sheath that is around there, okay? So let me see here. This area here, you're going to find the T cells, and then close, like on the border, close to your red pulp, you're going to find those follicles. Those areas is where your B cells like to hang around, okay? And it's not very much at the red pulp, but it's very close to it. Is vicinal, or is very close to your PALS area, periarterial sheath, because this is where the T cells are, and then this is where the B cells are. So you're going to have a place where they can interact with each other, okay? Now, at the end of those arterioles, or these little capillaries, you're going to find that you have open-ended capillaries, meaning that they are not going to be connected to the veins. When you look at the circulation, the blood here is not in a closed circulation. This is more like an open circulation. So the blood is going to spill out and leak out of those arteries and flood all of the zone. And this is why it's called the red zone. Okay, so let's see what happens here. You see, this is your artery, and at the end of the artery is going to be open-ended, and the blood is going to spill out. Okay, when the blood spills out, is going to be filtered over here, is going to interact with macrophages and other stuff, And then if the red blood cells are old or not functional or not bending well or tagged, they're going to be killed by macrophages. If the blood cell is healthy and flexible, it can squeeze through little openings and get back to the venous circulation, to the venous side. So the vein is going to be also part of this filter system, okay? So let me see here. All right, so here you have your white pulp, all right, the white pulp in the center. At the very edge of the white pulp is the margin of the white pulp, so you call that the marginal zone, okay? Here you have your periarteriolar lymphoid sheath, you call that pals. This is where your T cells hang around. Here you have your follicles, that's where your B cells hang around. And in this outer area, you have your macrophages that are going to clean up the blood. Oh, another question. That's also an easy question. The pals region of the spleen is populated by what type of cell? Some people are really awake. Answer very quickly. Okay, I'll give you 10 more seconds. Okay, anybody need more time? No? All right, so what's your answer? That's your B, that's your T cells. Okay. All right, so let me see what I remember. Oh, I still have a couple of minutes. I will take my minutes. All right, so here, where the blood spills out of your arteries and before it gets to the vein, is going to find a lot of filtrating type of structures, okay? And those you call the cords, okay? So here in this mesh, you're going to find a lot of macrophages that are hanging around in these cords, and they are ready to pick up damaged cells or viruses or stuff that can be in the blood, okay? So the red pulp is really what mainly makes the spleen. So you look here, like 75% of the spleen is made up of red pulp. So what is the percentage for your white pulp? Everybody awake? About 25 is right there, even if you're not awake. Okay. So a lot of red pulp. So what is happening here in the white pulp? Okay. So in the white pulp, you're going to have dendritic cells that are going to hang around. Okay. If they catch something, they're going to go and present to the T cells. Okay. T cells are going to be around here. T cells are here, right? Dendritic cells are hanging around. So they find something, they present to the T cells. T cells, if they get activated, they're going to go and interact with the B cells that are in the follicle. But at the same time, you know, the B cells are going to have to see the antigen, so they're going to be antigen experienced. Okay. Let me see what else I did not cover. This is about the same. Okay. So here is also a very nice schematics of what is happening. Here we have your white pulp, right? What kind of cells like to stay around here. Then you have the marginal zone and you have your red pulp, okay? And then what kind of cells like to hang around there. And here you have a nicer representation of what happens to the artery. And here is your white pulp, and then it spills out into the red pulp. There is open circulation here, and then it's going to be caught up by the venous side. Okay, so I also mentioned that. I'm going to stop here, and we pick up from this slide.